Okay. Thank you for the organizer. And I tried very hard for 6 hours on the highway in the airport, just couldn't make it to the destination. And so I really like to meet the people here. This is a group of people I hardly know, most of them. And so when I'm looking at the biological systems, I'm more from systems biology perspective, and the cell always, to me, is the most important unit and in biology. And basically, they have its own life and play a very important role, as all we know. So when we talk about the cells, in the old days, we classified them into groups, into types. If you go to read the textbook, right, about 5 to 10 years ago, if we train for MD, there are about 250 types of cells. And then we had a lot of discussions and knowledge about how cells will interact with each other and to give emerging properties. So only recently, about 8 years ago, the technology allow us to profile gene expression at individual cell level. Right? So basically, you granted a piece of tissue and then you put in a microfluidic device or some sort of device similar, and then you sort out individual cells and then you sequence it. Now, once you sequence it, then you start to look at it, right? So looking at it, and how you're looking at it. Okay, so when we're looking at those data, it basically is just one big matrix, right? In the vertical direction is genes, gene expression, information. In the horizontal direction is cells. How many cells? Tens of thousands now, but in the old days, means 4 or 5 years ago, usually it's in the range of hundreds and thousands. Because this is cells as identity as a function of genes, and the question becomes how you're looking at it, right? So first you use some kind of dimension reduction method, and you're looking at it, and then you color them and group them, and then that basically you give a name of each group of cells. So now we're looking at a low dimension and many 3 groups of cells. And then you're looking at the marker genes, means what are the important genes representing each group of cells. That's usually they call gene expression analysis, different gene expressions. And then they in the early days, means about 6, 7 years ago, and people come up with this idea, pseudo time, means arranging the cells in a low dimension based on the high dimensional gene expression similarity. The idea is if the vector of 2 vectors here are very similar to each other, means the gene dependence are similar to each other, the potentially they're very close to each other in terms of their cell lineages. So we also know the genomics data are very static, right? It just literally just a matrix. On the other hand, when we describe mechanism, we use the words spatiotemporal. We always say what causes what and what in which location gives rise to what, right? So in the world of physics, in particular for this audience, I think I'm pretty much the one standing between you and the dinner. And then when we're looking at a static image like this, we can almost imagine, right? And also in reality, we can even write down the equation, the Fluid's equation to describe those dynamics. But in biology, we don't have such well-defined, clear-cut first principle equation at the cellular level. There's many at the molecular level. And then the question is, use the limited or whatever knowledge available. How can we infer spatial temporal mechanisms? Okay, so that's the goal of my talk. And I will go shallow, but with a breadth to touch a few areas, because this is a fast evolving areas. So how much machine learning in my talk, and you will see. But most of them, it's not supervised learning. You can say it's more unsupervised. So I was first talk about how to infer from a static picture and to transitions. Now, the traditional language in the single cell world is the cells, clusters, developmental cell lineages, transitions. The question is, can we link this to a more dynamic system language? So right, so that's one. And then on this side, and then you can, once you become a more dynamic system language, then the things can potentially be more a wider box, more interpretable, right? So instead of giving you all the details, let me just outline. Basically, is we try to quote unquote fit the data into some dynamic system descriptions with the emphasize our attractors and the transitions. And I will not get into all the details. Basically, we consider the individual cells is zigzag particles, and the gene expression has noise, and then you can construct a graph. Then the traditional, we do not invent the diffusion map method, can give you some estimate on the transition probability between the cells. And once with the individual cells level information, you can cross-screen to different levels, right? So then you just, in a simple language, you almost like fit the data, but you don't fit the data with the explicit dynamic system with parameters. You fit the data with the attractors, with the saddles, okay? And then you can immediately connect the attractors and the saddles and in the form of almost like landscape. And then you can also compute in transition probability. So this 35% really, this 35.1 really means 35%, 0.3, 0.5 transitions. So this is an example of EMT. For many experts in this audience, it basically says that, that's an intermediate state, you can make a transition, you can quantify. So I will not get into the biology details. And then you can do the coarse grain. When the number of cells is big, at the end of the day, you actually don't care about individual cells. You care about a number of cells that give you distinct properties, right? So, and then we can do the coarse grain, for example, for this case, it's demonstration. You can roughly 4 cells as a state. You can see the micro state on the right and the individuals on the left has similar patterns, okay? This is another data set. And then you can track very complicated lineages. This is the blood cell systems. And then you can looking at large data sets and to do coarse grain and to analyze, in particular, to analyze complex cell lineage. So I quickly went through this construction of kind of a traditional landscape, right? Now the next is how can we more information, spatial information. So one of those cells talks to each other, right, through ligand receptor. So, however, here what we have is gene expression information. The data set I just described, it's only gene expression. And then can we use gene expression information to, for example, to model the protein information and then to looking at potential cell-cell talk. And we made the very first effort about 3 or 4 years ago. And if you think about it, the individual cell is almost like a sample of many cases. Then you can use this as a way to estimate, for example, the ligand receptor relationship, right? So if you use a correlated language, it's a very easy to understand. If the ligand information is high, receptors are high, the downstream genes are high, and in a statistical sense, and then potentially they talk to each other. So we develop at the individual cell level, cell-cell communications, okay? And then we found the accuracy is not as good, and then we move to more cell population, right? This is the more classic kinetic model and ligand receptors. That we model them. We can look at probabilities. We're looking at statistical significance. And in order to do that, we have also developed a huge database of about 2,000 pairs of ligand receptors. And then we use that model to infer. I skipped all the details. So once you're with the network, remember we have about 2,000 pairs of ligand receptors. Then you have many different ligand receptor interactions. Can you compare the network? So for example, we use more standard manifold learning for classifications. Many network analysis tools can be directly used. And then we publish the paper, and then for whatever reason, people use it a lot and highly cited. is one of the most highly cited Nature Communications paper during the past years. So let me just give you what we can do from such tools, right? So this is our self-work, our own work. It's looking at the old and the young wound. Okay, means the old mouse and the young mouse, you cut the wound, look at how it's healing. So that's looking at the ligand receptor interactions. Look at the age. You can see what immediately you can see is there's many different cell types, but don't get into the biology. So the first reaction, the old folks, the old mouse, the old wound actually has more synchronous. Okay. So what been surprised at the beginning? So basically, it's they're working very hard to communicate, but we do know the outcome, the wound for the old mouse is not as good as the young ones. Okay. And then we're also looking at the basal cell carcinoma, and then you can see that we can do comparison between the one with the cancer or without the cancer. You look at the signalings, in particular, look at the immune-related signalings. Look at here, the huge differences, right? The BCCs. Okay, so after our work, after our publications, many other high-profile or journal published the work using our method for COVID, for brains. Okay, so now that data set, you should immediately ask me, has no spatial information. Okay, and then we all know ligand receptor interaction most of the time has a strong spatial limits in terms of the length. So basically, if you have the data, which is the matrix, okay, and then you have the imaging with a few genes, and can you use that information to guide you analysis? So basically, can you do a map between these two data sets? Basically, here suppose there's 20 genes. Here, of course, is 20,000 genes. So you can do a map, right? You can map, of course, put it on certain constraints. The intracellular data structure has to be maintained in some ways. The inter-data set has to have some similarity, of course. And then you do all of this, it come out with something basically it's optimal transport, right? I don't want to get a mathematical detail. Basically, we use a combination of different optimal transport methods to obtain the map. So once with the map, and then you have spatial information now. So for example, this is Drosophila, there are 4 genes. This are published public data sets. You integrate it's 84 genes with spatial information. You can do much more rich analysis about how the information, for example, the wind signaling, the windless is corresponding to the wind, okay? In vertebrate, the DPP is corresponding to the BMP, and then you can see how they talk with different subgroup of cells. The zebrafish is similar. Then you can go then we actually tested, and we tested using the skin, and then we built a model. We're looking at the skin. We test the imaging data. We have about 20. We actually tested in the immune staining, basically it's protein information rather than the RNA information. That's where the method it depends on. Then you can apply this method to spatial transcription data. So it's an emerging technology. Now it's become more and more popular and useful and mature. Means those are the individual cells. Basically, individual cells you can measure in space. There's a single, very simple space, and then you can couple. You can each dot has 20-some thousand gene information. Okay, then you can analyze the communications. Now the question becomes, how you analyze the spatial imaging with a high dimensional information. So we all know for the imaging, for the classic color imaging, you basically have the 3 channel at each grid or a spot or whatever you want to say, the spatial resolution. And then now you have 20-some thousand genes. Then how did you classify them? How did you so-called high dimensional segmentation? So we developed a method called space flow. It's almost accepted. And then you can see, for example, that's ground truth. We compare it with many existing method. The space flow, the new method, clearly give something which is more close to the ground truth. Now, then you're asking how we do space flow. The bottom line information of our space flow methodology is to try to incorporate both the expression similarity and the spatial proximities, right? So basically, we use a deep graph informatic framework, and we put each dot, which could be cell, could be spot, into a graph, and then you do encoding, and then in a low dimensional space, you do regularization, you add in regularization. That's where we enforce this red two properties. Okay, so more specifically, get a little bit of math into it, because the traditional discriminant loss in this training method is something like that. And then we add in information, try to make the cell, when they're similar, they get into close to each other. When they're far, they are repelled to each other in the embedded regularization space. So Herbie, how much more time I have?
You should be finishing up.
Oh, okay. Let me just finishing up. Say, now there's so I give you a few examples of temporal and spatial. And then the data now becomes much more rich. There's many data sets from different labs. So then the machine learning techniques, as a quick goes, it's very important to how to distinguish as much as you can. Of course, that's impossible to do all of them, the technical variation and the biological variation. So this is the one we use non-negative matrix factorization way to learn the variabilities. Once you get into the RNA-seq, which is RNA information, and the chromatin accessibility, then these two data sets are mathematically fundamentally different. One is more continuous, dense in terms of info. Another one is more sparse binary. Then how to get those information mapped into the data? So we use iterative learning to basically map this into the same low dimensional space and analyze it. So let me just, then can you use the bulk data, right, to train, and then use the single cell data to learn, right, to learn the transcription factor binding information, and we made some effort and in that direction, and also try to looking at how the regulatory information flows using single cell information. This is some work just coming out about 2 weeks ago. So let me, since I just give you a broad kind of stroke, very quick and touched on a few areas. This is a fast evolving area, and what question you ask is even unclear. And we tried our best from the systems biology perspective to look at the genomics data, and we try to get the most out of them for the spatial temporal aspect of the data. And so let me thank the people who did all the work and also the support. And sorry, I get over a little bit. And thank you.
So I have a question about one of the techniques. Is it on? Okay. One of the techniques you used was the optimum transport. Was it just used as a technique to compare two distribution?
Yes.
Or is this some underlying, because he's talking about spatial,
Yes.
dependence of this information, I mean, transcriptome information. Is there a really underlying physical reason to use that optimum transport?
No, we just, the key is this data set is a distribution. This data set is a distribution, and then we connect them, the two distributions. Now you can add in more and more concepts. That's why the optimum transport we use, it's getting more and more complicated. It's basically, you can keep adding prior knowledge into it. Okay. And the physical constraints as possible. We have another major effort following up this week, this approach by adding more prior knowledge and physical constraints. For example, competition between two ligands and one receptor, one type of a receptor, right? That's cannot be directly incorporated in the existing optimal transport method. but we're developing it.
Follow up on the optimal transport one. So the prior knowledge goes in as different ground metrics, because I see a lot of different embellishments here, like unbalanced OT, structured OT. How do you put in the knowledge?
Oh, you basically just keep adding a lot of constraints.
So this is constraints in the
adding regularizations, right? That's the easiest way to do, right? But computationally, you know. And then very recently we started the effort in what do you call the dynamical, using the dynamic optimal transport to connect in the time. Then you become the more differential equations, and then the computation becomes more challenge, because you use the advantages, you have a continuous description. And the disadvantage is then you basically almost like fit the data into high dimensional convection reaction diffusion equations. Then you have to use some method to solve it. Then we use kind of a neural ODE. So that's another place to use in a deep learning method. We use neural ODE to solve it. Means you use neural ODE to represent that equations.
I see. I mean, one tiny comment I wanted to make is in these problems, I don't know about single cell data very much, but presumably it's easier to compare two things on the single cell sequencing part and compare to images of the spatial data. So people in NLP have developed variants of these things called Gromov versus Stein metrics, they are called.
Yeah, that's exactly what we're using. Yeah.
Okay, cool. Yeah.
Yeah. I just didn't give the details. Yeah. So now the challenge is to bring this to space. That's much challenging than before.
Any other general questions? We have some more general remarks that maybe you want to say. Okay. So